will go, if he's aware, Aya, he should go to this one because he never visited this one. Make sense, Aya, for your question? But the light one was once explicit as well. I don't understand, Ziad. But we will talk about the associative. Maybe it will be more clear. Okay? Don't stress about it. Oh, Sana, you're asking too much details. No. Don't worry about the emotional associated learning now. Okay? So let's go ahead. Guys, I'm glad I'm getting your attention, but let's move on. We will have more examples, okay? So just relax. We are associating the light with the cheese, with the wind, right? It's the Wednesday, like I said. So because over the 2 weeks of the training the animals, they learn to revisit only the arms they had the light. So this is the Wednesday task. In contrast to the wind shift, it's like there is a place where you can find the cheese. Next day or after 2 days, he will learn, oh, I visited this place, so there's no cheese here. So he's aware. Here in his background, he will always go where is the light. It's just like when you do the things without thinking. It's just like a habit, if you want to call it. Anyhow, let's go ahead and continue. Let's talk, guys, now a little bit more about the implicit memory and how can be associative or none. Okay, and this is that challenging. Let's giving an example. Let's start with our example. So what does it mean, non-associative learning? It means the animals will learn about the properties of a single stimulus. Remember, single stimulus, because when we talk later on about the associative one, you need 2 stimulus. Okay, so when you have a single stimulus, that's why you have, and that implicit memory, do you guys have 2 or 1 stimulus? For example, we have 2. We have the cheese and we have the light, not only 1 stimulus. Okay, but when you have a single stimulus, believe me or not, believe it. No, you have to believe me. I'm not going to give you the choice not to believe me, because the guy who discovered this, he won a Nobel Prize in physiology, and he did his experiment on a snail. And I'm using actually the figures from his book, Eric Kandel. You can Google it. Google him. I think he won it in 2000, so 21 years ago. Super talented neuroscientist. But there's 2 forms of the non-associative. Enough of the guy. Let's go back to the non-associative learning. There are 2 forms we have to know: the habituation and sensitization. Habituation. Let me give you an example. If you are sitting in your room, let's say, and you have very good working AC, and it's dropping water and making a small noise, at the point, you will just get used to it. Every single day, it's going to be dropping. Let's talk in the summer, of course. I'm not switching it in the winter, unless something wrong with your body. But anyhow, so if you have the habituation, it's just like a decrease in a response that occurs when a benign stimulus is presented repeatedly. Just like something, it's in your background. If you are sitting in your room and there's a construction every single day, till they finish that street or whatever. So you get it in your background, the habituation, because after time and time, this kind of learning is decreasing in the response because you get used to it. On the other side, when I mean the sensitization, okay, it's an enhanced response to a wide variety of stimuli after presentation of intense or noxious stimulus, which means strong stimulus. What does this mean? Look, This is the experiment has been done. This is habituation. You are touching the siphon for the snail, okay, or aplasia they call it. This is the control. When you touch it, look to the sensory neuron, interneurons, motor neurons. The gel will withdraw. But if you keep touching, I mean, of course, touching gently. You're not like tickling, let's say, the siphon part of the snail. You can see with time it's decreasing. So what reduced the effectiveness of the synaptic transmission between the sensory neurons and their postsynaptic cell? This is the sensory and the postsynaptic. What does cause this decrease actually? You know what? In a short story, this call it is the habituation results from the activity dependent, where you have a depression in the presynaptic site because you are getting used to it. So this reduction of the transmission or the neurotransmitter, you can see this is too much neurotransmitter, but this is a control. You do it one time. But if you keep touching it from time to time, habituation, this is a single stimulus again, you will just get used to it. And you just say, yeah, whatever, in your background. Yeah, whatever. I'm not going to be bothered by this AC. I'm not going to be bothered by, I need to stay focused on whatever I'm doing as a task. So this is an example of habituation. I hope it's clear. Okay, now let's talk about the sensitization. Sensitization, this is the one you are doing. If you're doing just the one, this side is the habituation. But what if you touch the tail, for example, and make it very noxious stimulus to the tail? You put it here instead of here and make it painful. I mean, we'll just make it painful. And this is what's called sensitization, because you are applying noxious, strong stimulus at this time, but in a different part of the body of the snail or the aplasia, whatever they call it. So the sensitization of the gel withdrawal reflex, and I always like to bold and highlight the important thing in the slide, make it easy for you. So look at here what I said. Sensitization of the gel withdrawal reflex is produced by applying noxious stimulus to another part of the body. Okay, so let's talk about this part here, the dashed square, and highlight what happened. Why this action? Because now the snail is think about there's an enemy or there's something dangerous in her surrounding media. So the withdrawal was significant, way significant if you want to compare to the habituation and even to the control, if you compare to the control. So what's going to happen? You guys, we've talked about this many times, I think, in my previous lectures. This is why I told you we will come back to it. And this is the time I told you about. This is the serotonin, the 5-HT. So the releasing of the, again, this is from this slide. So this is the motor neuron. This is the sensory one. And this is also the sensory, but from the tail, okay? And this is the interneuron. What's happening here? We have two pathways. We have two pathways. Remember what I told you before? When you have the GS, the S letter, so the G-coupled receptor protein. You guys remember what is this referred to, the S? You guys still remember? Or you forgot? Let's see your memory now. Oh my God, I need to open the chat. You guys remember cyclic AMP stimulation? Thank you, Sama. Yes, thank you, Maria. Stimulatory, Aya. Excellent, Bushra. So S for stimulatory, stimulation. What about the I? Remember, guys, right? The I for inhibition, correct? What about the Q? What about the Q? When you see the Q word. No. What is IP, calcium? Okay, you're close, Aya. Phosphorylation, excellent. That's what I need to hear. It means kinases, Muhammad. Excellent. The PLC. Well, you can read it, yeah. But the idea is phosphorylation. Okay. We're not gonna talk about which kinase, but in this case, yes, we will talk. So in this case, this is the pathway. I am not a person like to read everything on slide. Whatever is here is just very simple. You're activating the pathway 1, which is by the cyclic AMP, of course, by adenine cyclase. This means this is the PK, the protein kinase A. This protein kinase A, as you can see here, it will move, it will enhance the flow of the calcium in the channel and the phosphorylation of the potassium, the channel over here. Another pathway through the protein kinases, which is the DAG, we call it. Remember this guy, the diacylglycerol, and this guy will go through the protein kinase C, and this guy also parallel with the PKA. Oh my God, sorry. What's happening? Okay, so through the pathway 1, we have the cycling dependent through the PKA. That's all I want you to appreciate. Pathway 1 is the PKA. Pathway 2 is the PKC. How simple is that? Both of them, they will overlap on this one. But this one going through the adenine cyclase, this one is going through the PLC, DAG, and the PKC. You should know this already before. How awesome is that? And of course, you're releasing the glutamate that will bind to the corresponding receptors in the motor neuron. How simple? I'm not going to ask super details. All I need you to know these 2 pathways for this one, because it will come again and again later on. So let's talk about the associative one. The associative one, okay, 2 forms of the associative learning. There's the classical conditioning and the operant conditioning. So let's talk about first the classical one. Okay, very simple example. I always like to give examples. It might stick to your brain as a long-term memory, hopefully. The classical conditioning, it means it will involve, learning a relationship between 2 stimuli. One stimulus, we will call it conditioned stimulus, which we will refer to the CS. The unconditioned one is the, oh my God, sorry about that. The unconditioned one is the US. Now, the conditioned one means like a light, voice, touch. Okay? Remember that. Those 3, for example, light, you are hearing a tone or like a touch. The US, or the unconditioned stimulus, it's more reinforcement. Okay? More. It means a shock, heat shock, for example, or electric shock, or it could be a food. Okay? But it will, because it's normally produce a normal, strong, consistent to the response. That's mean, let's say, okay, let me give you an example for that. It will make more sense for you guys. Let's see. For the classical, I think I have a example for it later on. So, but for the unconditioned responses, they are innate because they are produced without learning. That's the difference between. But now the timing or the pairing between the CS and the US is very, very important. If you pair your conditioned stimulus with your unconditioned stimulus, okay, that means you are producing conditioned response. Unpairing them, it will not produce conditioned response. If you have a, let's say, if you have a dog, okay, maybe the dog is easier way. And the moment he smells the steak, you wanna give him a steak, like celebrating something, whatever. The moment the dog will see the steak or smell it, let's say, what would you expect for his saliva? Huh? It will go. Yeah, I have to open. Okay, increase salivations, right? There was more increase. He's hungry. He's excited about. So this kind of response or stimuli, we call it unconditioned or conditioned now. Unconditioned, right, Sarah? Okay, excellent. It's unconditioned, Bushra. Excellent. What about if you associate or pair that stimulus, which is the steak, with the, let's say, with the bell, or oh my gosh, or sorry, I don't know why it's going, with the whistle, okay, with the whistle, for example. So you are, so the whistle in this case, which is a voice, right? It's a condition. We agree about that, right, Walid and Abdullah? It's a condition. If you pair every time you whistle your dog and call him, I'll call her, whatever, and you introduce to him or introduce to the dog the meat, you would expect the same response, salivation. What if you do one of the time, if you keep this repeating this, and one of the time you didn't serve him the steak, and you only whistle him, would you expect the saliva, the salivation will come out also, the increase of the saliva? You guys agree about that? He learned. Exactly. Excellent, guys. He learned. This is associative learning. You are pairing this stimulus with this stimulus. Make sense now to you? Make sense? Okay? All clear, inshallah, for everybody? Okay, excellent. All right, let's move on. This is just a quick example for the what I said. This is the unpair, and you can see when you pair it, you do the same thing. This is now two stimulus. Okay, this is we're talking about the. Let me remove this somewhere. Okay, this is the unpaired pathway. Do you think you will have the same learning versus this one? This is the unassociated response, unconditioned response, and this is the conditioned one. Make sense one? This is before training, and this is after training. You can see significantly the gill motor neurons versus this one. You would expect when you have a pairing, you are teaching this snail to have a significant withdrawal with this one because you are pairing this with this. The moment you apply the pairing with them, it will have learning for this associated type of meta memory. Make sense, guys? Maybe the dog was more clear. Let's see another example. Here, another example, guys. And I brought this example specifically because the amygdala, we're going to talk about that tomorrow, inshallah, in the emotion lecture. The center of the fear is just like when you have, before your exam, maybe, let's say some of you, it will stimulate it because he is afraid from the exam. And I know everybody is that. I used to do that, the same thing. But what if we do, oh my God, I'm so sorry. I don't know why I keep changing. Okay. If you apply just classical music, Beethoven, whatever, for that small mouse, okay? Just, do you think his response will be scary? Just wait, Sarah. How about you give me let me finish this example. Maybe it will be more clear for you. Okay? Samiha raised her hand. You want to talk, Samiha? Is that what you mean? If you raise your hand? Okay, by mistake. Okay, thank you. So if you have just sound coming out to this poor guy, mouse here. And Sarah, just give me 1 minute, okay? Let me explain this, and we'll go back to your question to see if it's clear or not. And then, what do you think from his behavior? It's just like normal. If you check the electrophysiology, if you do the patch clamp, whatever, for his, it doesn't care, Walid. What's going on? It's just amusing. You're keeping it like every day, you're giving it. But what will happen, and remember that, let's be careful. Like, if you pair that voice immediately before you electric shock the mouse, immediately before, okay? If you keep doing this, pairing this with this, pairing immediately before, remember, not being separately, I'm saying pairing. This is why very important to understand that. If you are pairing it after several training for that mouse, what will happen, would you think, when you just produce that music or that tone or whatever that sound? He will be scared. Exactly, Sama. It's just like in a different pathway. This is very important to understanding. This is before conditioning, and this is after conditioning, and you can see significant change. And this is the amygdala. And later on, tomorrow, just let me remind you, please don't you send me an email. Is this the notes under the slide are important? Actually, I'm reading from them to explain to you. So everything in the presentation is required. Please don't send me an email at 3 a.m. Asking for these notes. There's not too much notes. It's explanation for the slide. Okay. So let's go back to this amygdala. Yes, Walid, excellent. But what I'm trying to tell you, the amygdala has different nucleuses, a lot more than one, lateral, central. Don't worry about it. I'm not going to ask you about details about this. All you need to know is just the amygdala. I don't go through details, which nucleus. This is the lateral one, I think, or the central one. So there's a lateral and central one in this experiment. So don't worry about it, okay? Specifically, what type of the nucleus and the amygdala that will do the excitatory or inhibitory? No, I don't ask this. This is not, it's going to be very hard. That's not type of questions I like to ask, okay? So amygdala is the center of fear response, like Walid mentioned. So far so good, guys. So look at this example. Very important example to know. Very important, okay? So if we have an experiment and we are testing the difference between the pairing and unpairing the conditioned stimulus, and in this case, the shock is the unconditioned stimulus. What would you expect if the more we pair, the strength of the conditioning? Look at here, 0% unpairing. So don't get confused, unpairing, 0%. So all of the stimulus in red, the green color, meaning unpaired shock. So they shock it here, but it's not paired with the sound, okay? It's not paired. It's only paired with the sound when it's red, for example. What would you expect the conditioning response? It will increase the strength with pairing or unpairing now, after all this explanation I did. Shock is unconditioned stimulus, Bushra. Remember, we said light, sound, touch is the conditioned one. Food, shock, noxious stimulus is the unconditioned one. Okay, Bushra, make sense? So we have conditioning. The strength of conditioning will increase dramatically if you pair both of them. Make sense, guys? So far, so good? Yes, Sama, it's with the pairing. The more pairing, the more learning, the more conditioning response for these animals. Like we said in the previous example, that poor mouse, when you just introduce the music to him, he was so scared. Same thing for your dog. When you whistle for him and you just lying to him and not giving him the steak, and after several attempts, you will know that he will not believe you, for example. And this is a different type of learning. Okay? So let's move on, guys. Now, oh my God, there's too much words in this slide, but very simple. Operant conditioning, take it from me and listen carefully to this. It's associated with your behavior, specific behavior. For example, my son, he make a big mess with the Play-Doh. Okay, I buy Play-Doh for him from Al-Hazaz because it's very cheap. Because it's very expensive if you want to buy him the original Play-Doh because he consumes a lot. So if he doesn't clean up, I will send him to timeout. This is my punishment. Okay, because the operant conditioning, guys, it can be 4 types. Okay, like positive reinforcement, positive punishment, negative reinforcement, or negative punishment. It has to be, okay, something you get this if you do this. You guys probably, maybe some of your parents will say, hey, high grades, you get the car. Low grades, you're out of the house. Maybe. Okay, this is an operant conditioning. Okay, Walid, call it operant, so we don't our parents don't get offended. So operant conditioning involved a learning and the relationship between the animal, for example, behavior, and the consequences of that behavior. Or for a human, for example, you are, let's say, I give an example. Hey, Malik, if you clean up, you get more Play-Doh. That's a positive, right? You are encouraging, and this is type of learning. You clean up, you will get more Play-Doh. You don't clean up, you will be punished. It's not like so. Make sense? So, Ala, how to differentiate between condition and condition? Oh, come on, man. I already explained condition and unconditioned. What do you mean condition and condition? You mean the stimulus? Are you talking about the stimulus, Ala? I hope I'm saying your name right. Well, he is not answering. Okay. Is it clear for you guys, the operant conditioning? Anybody has a misunderstanding? Please, so we can move on. It's very clear. It's between the consequences of your behavior. Okay, we'll decide. It's very simple. Okay, so let's go ahead and move. Close the chat. So now let's talk about the storage of implicit memory and what's going to happen to the synapses because of that. Remember, this is very important, what I said here. Long-term storage of implicit memory involves changes in chromatin structure and gene expression mediated by the cyclic AMP, protein kinase A, and the CRIP. And we're going to talk about this. This is the long-term storage, okay, and of the implicit. We're going to talk about the explicit also. For the long term, we're going to talk about the implicit and the explicit, but I separated them so you guys don't get confused. So this is a very good example and a very good experiment I put for you guys. I added long-term habituation lead to lose to the synapse. Remember, the concept that Eric Kandel, he won his Nobel Prize in a very, I can summarize the whole lectures in one thing, in one short sentence. Memory is stored in synapses. If you form more synapses, you are forming long-term memory, long-term storage. The more synapses you are, meaning the more sensitization, meaning you are giving more action potential, meaning more release of a neurotransmitter. That will change the gene eventually expression and more synapses will form. So look at this here, for example, this experiment. Long-term anatomical changes. This is the control, habituated. Habituated, actually, it's the opposite. It's kind of a depression. But in the sensitization, you will have increased the number of synapses, as you can see here. Okay? So the number of the synaptic or synapses formation in this sensory neuron is increased due to the sensitization. And remember, sensitization stimulus is a noxious, a strong one. Habituation is not. And that's why, like I give the example for the drop of the water from the AC, for example, in your room, you get used to it. Okay? And this is the control. Make sense, guys, for this term? Because I'm going to talk about the structure inside the cell, what's going to happen. It's supposed to be pretty much clear. Okay? So let's talk about what's going to happen at the cellular level for this one. Look at this one. Oh my goodness, this is too busy, but let's make it very simple. Remember that slide, and we said about what's going to happen on this graph. This is the big one. Okay? We're going to talk, remember this pathway? We talked about it, and we have a pathway 1 and pathway 2 when we talked about the sensitization. But what will happen if you have a long-term sensitization? It stays for a long time. You are forming a memory in this line. This is why you have more synapses. This is why we have more synapses. More synapses, a growth of a new synaptic connection. You see that? We didn't have that one in just a simple sensitization. Okay? But if you keep it for a long time, so Bushra is saying more synapses, more storage, long-term memory. You're right. But let's talk about it more details now, guys. Okay? So what's going to happen at the terms, sorry. So we've talked about this pathway. The cyclic AMP will activate the protein kinase A, and this will enhance the release of the neurotransmitter to the synaptic cleft. We don't want to go ahead and repeat that. We've talked about this. But let's talk about at the cellular level, what's going to happen here. As you can see in the pathway number 1, the MAP kinase is the master guy to go to the nucleus and affect the crypt. So what is this? What's going on here? In this case, the histones, it's the transcription block. You have to release it and change the structure of the chromatin in order the transcription to be accessible. What does this mean? This is the basal state, for example, the CRIP1 or CRIP2, and this is the CRE. Okay, so in that case, for example, and I wrote all the abbreviation, guys, for you and under the slide. So, for example, the CRE, it's a cyclic AMP recognition element. The mouse. The CRE is the cyclic AMP recognition element. Okay. And what else? You will find a lot like poly2, like polymerase, RNA polymerase. Okay. So what's going to happen now? Let's talk about it. When you have the 5H, which is the serotonin, produce modification chromatin structure, the CRIP1 phosphorylation, remember, will cause the serotonin activate the protein kinase A, and this will phosphorylate the CRIP1, which is indirectly also will enhance CRIP2. Okay, and this is also induced by the MAP kinase I showed you in the previous one. What does that mean? You are allowing the CRIP1 to form a complex at the promoter. Now you relax the histone. You are changing the structure of the gene. Okay, and this cause binding of the CRIP to form a complex at the promoter with the CRIP binding protein, which is the CBP. Now the activated CRIP binding protein, this will cause acetylation for specific lysine residue. I'm not going to ask you details about this, okay? But I want you to understand the concept for this specific slide, how this will take a place to change the structure of the gene. This is a physiology part, okay? So we cannot change it, but there's acetylation, and this is the Tata box, very well known, but this will cause will recruit the RNA polymerase to that site, okay, because you are making a changes and reposition to the polymerase 2 and to be recruited to the initiation of the transcription site, okay? So I want you to, what is very important in this pathway, listen to this carefully. MAP kinase, phosphorylation for the CRIP2, CRIP1, then you activate transcription. Fair enough? Anybody want to look for more details? I already explained it. So you have the MAP kinase involved in the phosphorylation.
ما هو كنا جبنا العيد غشينا وما غشينا جبنا العيد.
Sorry guys,
أمي وأختي كانوا جنب بعض عادي.
Can you guys mute yourself?
I'm so sorry, doctor. I will mute her. Done.
Okay. All right. Yeah, yeah. So, okay. My short memory is affected now. Okay. Now, let's go back. You have a MAP kinase activated here in pathway 1. When you see MAP kinase, it's a kinase. That means phosphorylation. That means the CRIP1, CRIP2 were phosphorylated and take a place and initiate the transcription. Now, this means the ubiquitin hydrolase will be formed, and this will cause the degradation of the pKa regulatory subunit. That means persistent activity of the pKa, and that's what you need to know here. Part 5, the pathway for 5, and the pathway number 5, a growth of new synaptic connections. This is why when you have a long-term sensitization, you are forming new synaptic connections. And if you have habituation, okay, long-term habituation, it's the opposite. This will disappear actually. Make sense, guys? If you have a strong, noxious stimulus, like in the sensitization case, you are reaching the action potential. You are releasing the neurotransmitter. You keeping changing the structure of the gene, the chromatin structure, gene expression. That means growth. The synaptic connections will grow more and more. Make sense? This pathway? It's very easy. This one is fair enough to explain it very well. Can you please repeat what happened after transcription initiated? Okay. The transcription, after the transcription initiated, meaning the gene expression initiated, correct? So far, so good. When you have the initiation of the transcription, you are activating the gene transcription, as in pathway 3, for example. The protein kinase A also will activate the mitogen activated protein, the MAP or the MAP kinase. You can see it, okay? And why does this keep disappearing? Wow. And you have also the pathway 4, okay? The pathway number 4, where you have the CRIP, for example, 1 will encode for a ubiquitin hydrolase, a component of a specific ubiquitin proteasome. This will cause a cleavage of the regulatory element. Hydrolase, it means, sorry. Hydrolase, it means you are breaking apart the PK regulatory subunit, meaning persistent, more active protein kinase A. That's the idea. You transcribed the genes and initiated the transcription, meaning you have more ubiquitin hydrolase, meaning you are degrading the pKa regulatory subunit, meaning more persistent activity of the pKa, and that will enhance the release of the neurotransmitters. Okay, make sense? Ala, sensitization, not any sensitization, long-term sensitization, long-term sensitization. Okay. Sama, doctor, so the cyclic AMP will break the bond between, I didn't say the cyclic AMP. Cyclic AMP will initiate persistent activity. Yes, Yes, if you want to say it in a different way, the hydrolase will cause the degradation of regulatory element. PKA activate CREP and MAP kinase. Okay. Then push. So guys, look, it's been, or we have more. It's very clear. If you just read it, this one, it's very described, but I already mentioned it. I will just mention it one more time. And I can mention it through the steps very clearly here. If you have a repeated tail shock, and that means long-term sensitization, okay? Long-term sensitization. It's lasting for 1 or more days. So this will increase the cyclic AMP, and that means it will have an effect, the cyclic AMP, because it's a stimulatory effect. As you can, more protein will be synthesized. This mechanism is initiated by translocation of the protein kinase A to the nucleus, which is the pathway 1, okay? And also the MAP kinase, okay? You have the MAP kinase and you have the protein kinase A. The protein kinase A will phosphorylate the transcription activator CREP, which is, they call it the cyclic AMP response element, or the CREP1, it means the cyclic AMP response element binding protein 1. And also this is in pathway 2, okay? This is 1, and where's number 2? Here. This is pathway 2, okay? And if you go to the pathway 3, which is through 1, this one, same thing, but for the CREP1. We've talked about CREP2, and this is CREP1. Meaning the protein kinase A will also activate the I forgot to mention, this is the mitogen activated protein kinase A. So protein kinase A is working on 2 pathways, through the nucleus or through the protein to the MAPK kinase. That's just, by the way, it's good to pay to my attention. If you want to write the abbreviation, I'm fine with it. Okay? If you want to write the abbreviation, I'm okay with that. When you do the gene transcription or changing in the gene, that means you will release the ubiquitin hydrolase, okay, which is in the pathway 4, okay? And also you will synthesize a lot of protein that cause the growth of the new synaptic connection. That's it. I don't want to ask for too much details more than this in slide. That's all I need you to understand, okay? Protein kinase A, nucleus, and through the MAP kinase here, phosphorylate 2 and 1 for the CREP. You have hydrolase, the ubiquitin. This causes degradation for the regulatory element subunit, meaning persistent activity. On the other side, you have a growth of the new connection. So far, so good? You guys still here, right? I hope so. Okay. So I put this slide twice, so don't get scared. It's the same slide, just because I was referring to this. So this is why when you have your ubiquitin hydrolase, when you have persistent protein kinase A, when you have persistent activity of protein kinase A, you have a long-term sensitization because you are forming a new synapse. It's the opposite when you have a long-term habituation. Make sense? This is the same slide. 2 slides repeated, it's just the same one. It's just like to stick it in your brain, hopefully. Now let's go to the explicit memory. Okay, and we talked about the explicit 4 stages: encoding, storage, consolidation, retrieval. Now, why this is in red? Because in consolidation is where you change the gene, where you have a gene transcription, where you have a gene expression. That's why I bolded in red. So consolidation, now I cannot say the word right, involve expression of the gene and the protein synthesis that give a structural change in the synapse. So you have to reach, if you are in this stage now, you are storing long-term memory. Okay, so yes, yes, Ghadir, yes. Okay, so when you are having having more synapses, Ghadir, more synapses, they will have a neurotransmitter eventually, right? So retrieval is just a process where you store the information, you bring it back. Make sense? So 4 stages: encoding, you have the new information, you are just reading it; storage, the mechanism, when you want to retain the information; consolidation, where it's reach a stage where involve the gene expression and the protein synthesis to form what? To form what? What, guys? Huh? When you have a consolidation, what's you gonna form when you have a gene expression? Come on, synapses. Yes, it's a long memory, but it's a synapses. You remember, I summarized the lecture in one or a few words or one sentence. More synapse. I hope you guys are forming more synapse now. More synapse, more memory. Okay. Anyhow, let's go back to our dramatic story. Just like for the patient Henry, this is what he was missing. Remember, he was not able to make this information. He was not aware. His doctor, she will come to his. This is the part was removed and bilateral, and this is why you appreciate the importance of this structure. So let's talk about this as a very important. This pathway is very, very important. Why it's very important? Because the hippocampal synaptic circuit is very important for the awareness and the conscious memory. And for that, I want to show you our highlight here for this structure here. Let's go back to the laser. You have direct, indirect. Okay, so this is from the entorhinal so excuse me, entorhinal cortex. So this is the perforant pathway. So you have a direct and indirect. Just a second. You have a direct and trisynaptic. Why it's trisynaptic? Because you can see 1, 2, 3, trisynaptic. And this is the dentate region. This is the mossy fiber pathway. This is the Shaffer collateral pathway. You have to know this pathway. You have a direct one and the indirect. The indirect through the trisynaptic. This is the granule cell. This is the mossy fiber. And this is the Shaffer collateral pathway. So what's going to happen here, guys? They call it the trisynaptic or indirect, some textbook. Differentiate between these 3 pathways. The long-term potentiation, this is where the long-term storage for your explicit memory will happen. In the Shaffer collateral pathway is associative. Why we call it associative? Because simply it's associated with the NMDA receptor. If you block the NMDA receptor, as if you use the AVP drug, you will see complete block for the potentiation. There's no action potential generated. That means this pathway, this is why I said it in bold here, the LTP, long-term potentiation, requires activation of the NMDA receptor, meaning associative. That's it. So far, so good. It's not that hard, right? So if somebody asks you, it's me actually, not somebody. The long-term potentiation in the Shaffer collateral pathway, it's associative. Why? Because it requires the activation of the NMDA receptor. How you prove that? You block the NMDA receptor, AVP, you don't see any action. So far, so good. The second one is the direct perforant pathway, LTP. This guy is, they don't say it associative or non-associated because we have the LTP is dependent on the NMDA and partially on the L-type calcium channel. So both things. So you require an NMDA receptor and L-type calcium channel. That's why if you use the AVP to block the NMDA, you still have a signal. You still have EPSP. But if you block both of them, okay, that means both of them are required. Let's see the chat. Somebody had a question. Okay, so associative, this is for the long-term, Bushra, this is for the long-term potentiation. This is for your memory, the explicit memory, not any memory. It's just for explicit memory. How do you store your explicit memory? And remember, explicit memory like event, facts. For example, if you guys, so the Lakers, they won the championship 2020. Tampa Bay, the Bucks, they were the champions for the Super Bowl. It's a fact. So 2020 is a Corona year, if you want to call it, unfortunately. So this is a long-term potentiation. You are aware when you say these facts, these events, okay? This is explicit memory. You have it in your, after several years, we'll ask you, you are aware of this, okay? Potentiation, it's action potential. That's what I mean, potentiation. It means action potential, releasing of neurotransmitter. It's a synapse, remember? You have a synaptic terminal here. Makes sense, Bushra? You disappeared. Okay, thank you. So let's move on. So we have associated pathway, which is dependent on what? The first one, the Shaffer collateral pathway. Dependent on what, guys? Come on, show me your memory. Explicit memory, NMDA. Thank you, Abdullah. You saved them. What about the let me move one slide. What about the so you can you can see this other the perforant pathway, the direct perforant pathway. You need excellent Sama. You need the L-type channel of the L-type calcium channel, which is partially dependent. So blocking the NMDA receptors is not enough. Okay, two things. The L, this is all I'm asking to you guys to know. Okay, the L-type calcium channel and the NMDA receptors. But in the associative one, only NMDA receptors. This guy here, the mossy fiber, okay, the third and the last pathway is non-associative. I don't care about the NMDA receptor. Why? Because, sorry. Why? Because it doesn't require an NMDA receptor, because it requires protein kinase A. How easy is that? You block the protein kinase. I'm so sorry, my hand moves so fast. You block the protein kinase A by the H89, for example, drug inhibitor. You don't have potentiation, long-term potentiation, action potential generated through that one. Make sense? Three pathway: associative, depending on the NMDA receptor, okay? The perforant one, that dependent on the NMDA, and what we say, the L-type calcium channel, and the last one, the non-associative one, which is through the mossy fiber, okay? It's through the protein kinase A. That's all the story for the explicit memory and the part of the hippocampus. And that guy, Henry, the patient, the HM patient, he was missing that portion. So he will never, ever build this kind of type of memory. He will never remember his physician, Dr. Milner. She has to introduce herself every single day to him. Okay? There's no, so this is the Hebb's rule. Let's talk about it in bold. It means when you have an axon of a cell A excites cell B, repeating and persistent, and keep exciting it, okay? It means potentiation. Another way, for example, and it takes part in firing, some growth process or metabolic change take place in one or both cells. That means forming what, guys? Forming what? Come on. Synapses. Thank you. Thank you. Excellent. Some people still alive and surviving with me. I'm very sorry, but I'm trying to describe everything. I want to make it very clear for everyone. So this is how it starts. Okay. So what is the steps for this Hibs rule? If you have during normal low frequency, the synaptic transmission of the glutamate, for example, it will be released. Okay. And it will go, this is through the, for example, I mean, this is the Shaffer collateral axon. We're going to talk about now the Shaffer collateral axon. And this, when we talked about this, that means it's dependent on the what? The NMDA receptor. Remember? Because it's associative. Then it will act on the NMDA receptors. But the opening of the NMDA receptors, guys, unlike the AMP, the AMPA receptors, okay? Because first, the ionic, this receptor, which is like, it's not a G-coupled receptor, it's an ionotropic receptor, the glutamate must bind to the NMDA, and this will open the channel, and then this will change the resting membrane potential, depolarization, okay? And then this will be sufficient for the NMDA receptor to conduct ions because you are removing, of course, the magnesium from the receptor, okay? Then this will recruit eventually this guy over here to the cell membrane. This is how it starts. Then when you have induction of long-term potentiation, the previous one is just normal. When you have a long induction, long-term potentiation, more stuff will take an action. You have the calcium, the calmodulin. Remember this pathway. This is very important. The calcium, calmodulin, will have 4 pathways here. CAMK2, nitric oxide synthase, tyrosine kinase, protein kinase C. 4 steps by this guy. You can appreciate the calcium is the key factor for this long-term potentiation because it will affect the CAMK2, which also will have affect the AMPA, and the nitric oxide, which has a, remember when we said it's a neurotransmitter before, and it go back, let's call it retrograde signal. It will go back to the presynaptic, release more. And this will go through the protein kinase C, phosphorylate this physical, and go back to the cell, fuse to the cell membrane. That means in the, sorry, the expression of the long-term potentiation, you will have insertion of more AMPA receptors, meaning more actions taking place. So what's going to happen eventually? This is the Hib rules again. There's keep exciting A and B, meaning from silent synapse, meaning unsilent synapse. And they can be different. So for example, this is the normal one, no long-term potentiation. Cooperative, all of them, they are weak. All these signals are weak, but at the same time, they form, all of them, a long-term potentiation. Some of them, one of them is weak, inactive, or weak active. They still have a long-term potentiation. Of course, when you have 2, then you will have a strong effect, active input. So the last stage, you have, we've talked about this. I know I keep repeating slide, but this is good for you to slide and reduce it. So you have the calmodulin taking place on 4 parts, PKC, tyrosine kinase, and nitric oxide, the CAMK2. But what is very important, this is for the late, this is for the early LTP. But what about the LTP, the late LTP? There's a very important protein. It's PKM zeta. this symbol here, zeta, Z-E-T-A. I wrote it down. So this PKM, zeta, is very important for the LTP. And this guy will go cause formation of a new synapse. Of course, you have the calmodulin through the cyclic AMP, and then with the protein kinase A, we've talked about this. We'll go to the nucleus, effectors, okay, and will form. But this is very important step here. In the early LTP, you have this model going here. The calmodulin affect these four pathways, but at the same time, the calcium, the calmodulin pathway will affect on the late one with the help with the PKM zeta. Make sense? And then you will have formation of a new synapse. Very quick. I think this is the last 2 slides. Okay. If you have long-term depression, the opposite, low frequency, low calcium, what's going to happen eventually? Long-term depression, just the opposite, okay? Okay, it will have, sorry, keeps changing. Okay, low frequency, it means calcium is low, and this means, look at low concentration of calcium is thought to be insufficient to activate the CAMK2. So what is the key here? CAMK2. When you don't activate the CAMK2, the enzyme implicated of the LTP, rather LTD, is thought to result from the activation of the calcium-dependent phosphatase. Okay, and I honestly, I want you to understand when you the key in this one, for example, the difference between both of them. I will check the chat very quick. I will answer you guys. I will give you time. Oh my God, this is the last 2 slides. I will go through them, and that's it. The difference, I wrote this small difference between the LTP versus LTD. Here you have, oh my God, I'm very sorry about that. The LTP, I'm not gonna touch the keyboard or the mouse. And the LTP, okay, versus the LTD, okay, here you have increased synaptic efficacy. And the LTD, you are weakening the strength following long term. So low frequency stimulation of the afferent fibers, and the LTP, you have high frequency of the stimulation of afferent fibers. In LTP, you have high post-synaptic calcium. In LTD, like I explained, low post-synaptic calcium. And that means activation of the intracellular kinase, because you have really bother me. Maybe this will change. When you have a high post-synaptic calcium, you are activating the CAM kinase 2. When you have low post-synaptic, okay, you are activating several different things. And I forgot to mention the intracellular phosphatase calcineurin, okay, not the CAMK2. The calcineurin is different than the CAMK2. You activate this guy here, you activate this guy here. This is depression. This is what? Potentiation. Long-term potentiation, long-term depression. Here you will insert AMPA receptors at the postsynaptic site. Here you will remove the AMPA receptors. Okay, so makes sense? So the last slide now will test your knowledge. We've talked about the amygdala as the center of the fear in the brain, correct? Okay. So there is, if you have, we will talk about this more tomorrow. So I'm mentioning here today, and I'm gonna bring it tomorrow because it's about emotion also. It's related. But remember, the amygdala is the center of fear, and it's when they did a study for the patient where they have a bilateral damage to the amygdala or the hippocampus, they will have a different consequences on the implicit or the explicit memory, right? Because the amygdala is part of what? Implicit or explicit when we talked about it in that figure? Implicit. Excellent. And the hippocampus is in the hippocampus is part of the explicit memory. Excellent. Okay. Now the question. The patient who had a damage to their amygdala, they will show no physiological response to the CS, which means conditioned stimulus, indicating no implicit learning. Does that make sense for you guys? You damage the amygdala. If you expose them to conditioned stimulus, okay? No physiological response to condition because there's no implicit memory, but they have a good memory of conditioning experience because of the explicit memory in their background, what they had in the past. But if you damage it, they will not be able to build a new explicit memory if you damage the hippocampus. If you damage the amygdala, okay, then you are indicating no implicit learning because the amygdala is part of the implicit memory. What if you damage the hippocampus? And we know a very good example. It's our patient, Henry, that where he had that surgery and damaged, and the hippocampus and the amygdala and was all removed. But in this case, what if you only target the hippocampus? Okay, they will respond exactly, Abdullah. No explicit memory to build it, but they will respond normally to the condition stimulus, but have no conscious to the memory of the condition experience. It's just like the guy who's building, drawing the star. He doesn't remember, but he's repeating the task every single day, okay? Because it's a priming, it's a habit, it's a skills he's developing in his background, but he will not be aware every single day. You remind him about this. So for that patient. So do you guys have any questions? Hisham is asking, the person with damaged amygdala will have no physiological response, but will respond conscious because he remembers. Only damage, remember? Hisham, we're gonna talk about an example for a patient, and I will show you her MRI tomorrow. She's failing to express her fear. She can be happy, she can be sad, she can be excited, except being, if she watch a horror movie, she doesn't care because her amygdala is damaged. And amygdala, remember, is part of the implicit memory. Okay, but she's conscious about what is going on, but she will never express fear. I hope that makes sense. But tomorrow we'll talk more about that. You guys have any questions? And I'm very, very sorry. I promised to finish before. It's 4:48. I can see a lot of people left. They don't want to hear more of the memory. What determines if B is associative, cooperative, or specific? Where the old memory stored? Where is the old fear memory stored? Thank you, doctor. Okay. For the storage of the memory, it will be in a different part of the brain. And this part and this kind of questions is still under research. I don't want you to worry about this. I'm not going to ask you this question because this is based on research and publication, and it can be true and it cannot be true. But So don't worry, I'm not going to ask you where is the bad memory, old fear memory, old memory can be stored. But if you are asking about the type of the memory, like a fact or event, okay, we know it's an explicit memory, and we know it's in the medial, temporal medial loop. But if you're talking about implicit memory, we know where it's is stored depending on what type of the memory. If you go back, I should go back. If you go back, let's see, to that graph, it's a very nice summary in that graph. You guys can still see that. I don't know, it's like this one. If talking about this type of memory, okay, this is the implicit memory. This is the part are responsible for this implicit memory. The reflex pathway for the non-associative learning, habituation, sensitization we talked about. The fear for the amygdala. This is where your implicit non-declarative memory. So, but there are some research actually, not only in one specific place. So I would be careful. But your striatum, for example, is important for the procedures and the skills and the habits, priming in the new cortex, for example. And for the facts and the events, the anniversary, your birthday, like I said, the Lakers 2020, or the Super Bowl, these kind of events and facts, and the medial temporal lobe is responsible for that declarative or explicit memory. So I have a question. What determines the I don't understand what do you mean. What determines the LTP is associative, cooperative, or specific? I don't understand what do you mean. There are 3 pathways. Every pathway in the hippocampus, I'm expecting you're talking about the, let me see. The explicit memory, if I'm not wrong, this one. Okay. You have different pathway. It will go through the pathway, different pathway, not only specific pathway, but there's a direct one.
Doctor, I was curious because in the long-term potentiation, you put like different ones. Like there was cooperativity, and then there was like
yes.
So I was wondering like what determines if something is like long-term, but it's specific, or long-term cooperative. You know what I mean?
No.
Like it's down. It's in the slide. It's one of the final slides that you put.
More, go down. The memory, emotion.
Abel, before that. Another one, and another one. Yeah, the slide above that. Yeah,
this one.
Next, yeah, no, yes, this one.
No. Okay, I'm sorry. What I try to explain here. Do you have like different pathways? If you can, sometimes if you have a very weak signal, this is the Hebbian's root. This book was written in 1949, I think. And this is what Eric Kandel, he worked and he published that work in a neuron journal, which is a very highly prestigious journal. That's why he was awarded that Nobel Prize. He showed that either if you have a weak signal, then you don't have a LTP, no potentiation, no aggregation. But if you have a couple neurons together, for example, separately, having a weak signal, yes, it could be eventually an active input enough to initiate the the potentiation, let's say, the long-term potentiation. But it will be even better if you have one of them strong and one of them weak. It depends on the signaling through the neurons. Does that make sense? Because the LTP can be induced either by strong tetanic stimulation of a single pathway to a synapse, or by cooperative via weaker stimulation of many. So it depends on which pathway they go inside your body. So when one pathway into the synapse is sufficient, it's actually insufficient for the postsynaptic depolarization to induce the LTP.
Thank you so much. That's clear now. Thank you.
Seriously, is it clear? Seriously, I can explain it in a different way.
No, بالعكس. Thank you so much.
Okay, I'm so sorry, guys. I kept you for 2 hours. I'm really sorry, but I hope I'm doing just my best to explain everything, because I don't think it's that simple. The memory is kind of confusing. Giving more examples make it easy. You know, if either it's always stick in your brain when you have it to your with an example, live example. Do you guys have any questions? Feel free. Okay, this type of questions might take long. If you want to stop by my office, feel free anytime to stop by. You know, this week we are busy in the poster. So next week, because I'm a group leader and I have to see all the posters. Next week, I will be happy to see anybody in my office, sit down and explain the slides. Just wait for the emotion tomorrow before you start shooting me questions. And let me remind you something very important. Sleeping is very, very important for your memory. Even taking a nap is very, very important. I am a neuroscientist, and I can tell you, okay, sleeping is very, very important. This is an advice from me to you guys, because the memory, like I said, acquisition, consolidation, and the recall, all of them will be enhanced, especially the consolidation, where you have the gene expression, will be enhanced dramatically by sleeping, even taking a nap. So the students, I'm surprised some of them, they send me an email at 2 a.m., 3 a.m. asking questions. You guys, you need to rest your cells and inside your brain. Yes, my advice and my long-term memory, Sama, exactly. So the sleep does more help to sharpen the mind, actually. And the studies show that sleep affects the physical reflexes, fine motor skills, and judgment too. And there's a very good study show that participants who were lacking sleep were more likely to think they are right when they were in fact wrong. So I am Mr. Right, for example. It's an interesting study. And also the studies they did in the memory test, after single night of sleep or even a nap, they perform way much better in their task and their performance and their workout or their athletic field or whatever. So if they are athletic, for example, sleeping is very important for your memory, guys. I encourage you to, as a medical student, and inshallah I see all of you as a future doctor, to take care of that part and organize your sleeping time. Again, I'm very sorry. I took not full 2 hours, okay? Because I'm just giving my opinion about sleeping, how it's important. I'm not gonna ask you about that in the exam. Any questions, guys? I guess most of you, 50% left almost. So just, you guys are very tired. I'm also tired. Any question, guys? Seriously. Yes, Sana, take a nap now and you will memorize all these stuff I told you. Everybody take a nap, including me, if my kids let me sleep. Anybody want to have oversleeping good or bad? Oversleeping is not good, Sham. Come on, oversleeping. You're now taking my words for excuse to oversleep. Oh no, Sarah, definitely no. 4 hours? No, that's not enough. they said neuroscientists between 7 and 8 for your age. All of you are young, not old like me, but 7 and 8 hours. 4 hours, no. You will never perform very well. And this is published data. Anybody have any concern, any questions? I sleep 9 to 12. Well, you're lucky, Sham. I wish I can do that. I'm a morning person, so 9 to 12 hours. 9 is good, yeah. 12, yeah. I don't know about the 12, honestly, Sham. You're a young guy. Any question, guys? Before we end the session, because Hala, she wanna go home. We are keeping her. Any question?
Thank you so much, doctor. This was very interesting.
Thank you, and I'm very sorry to keep you late. Very sorry, and thank you, guys. Okay, have a good evening. Assalamu alaikum, guys.